Good afternoon, dear all. I'm MOUIZZ Salaou, MSc graduated in Biotechnology and Plant Breeding from the Faculty of Agronomic Sciences of the University of Abomey-Calavi. I'm a research assistant working in the Molecular Biology Unit of the Laboratory of Genetics, Biotechnology and Seed Science. My pleasure to be with you for this talk entitled Collection of Genetic Resources of Selected Traditional African Vegetables in Agroecological Zones of Benin. After the introductions, I will talk about the objectives, material and methods, the results, and I will end this presentation by the perspectives. In tropical countries, there is a wide variety of edible plant species that can significantly contribute to food security and human health. Among those species, we have vegetables that are more diverse, colorful, and tasteful. They are strategic for reaching balanced diets and contribute in reducing the incidence of severe health ailments. Traditional African vegetables are particularly important for local communities because they are easily cultivable, they are affordable source of nutrients, they are adapted to local environmental constraints, and are used in traditional medicine. So they constitute key assets to support more nutrition-sensitive agriculture in the context of climate change. Despite their importance, traditional African vegetables are underutilized by farmers and are neglected by research and development programs. This led to the loss of the traditional knowledge and the genetic erosion of local land races and populations. Moreover, accessions in World Genebank are composed of more grain, pearls, and fiber with a small number of traditional African vegetables. It is therefore important to take actions to adequately collect and conserve food diversity within the gene pools. Recent studies have shown that Benin and South Cameroon are hotspots of diversity of species, respectively in the West Tropical Africa and the West Tropical Central Africa. Moreover, vegetables' genetic resources in those countries are least represented in ex situ collections. The authors have then concluded that Benin and Cameroon are priority countries for germplasm collection. The objective of this work was to ensure the conservation of the available genetic diversity of some traditional African vegetables in Benin. Specifically, it aimed at collecting accessions of 18 traditional African vegetables in the 8 agroecological zones of Benin and to regenerate and send the duplicates to WorldBird's genebank in Tanzania. The germplasm collection was carried out from July 14th to 29th, 2020 by 8 research assistants of the laboratory. 24 districts were selected as potential areas to collect based on the result of Ajidangako. The collecting form was crafted and uploaded on smartphone using Kobo Collect's application. A prior informed consent was signed by all the participating respondents. The respondents were selected using the snowball sampling methods. Around 217 villages and more than 470 farmers were surveyed. The figure 1 presents a map of the collecting area of those staffs. The collecting form was composed of the accession code, the location of the collected accession, the seed origin, the species uses by the provider, the local name of the landrace, and the name and contact of the supplier. The accession code was composed of the initials of the scientific name, the first three letters of a district, and the sample number of the accession. So for instance, MCDJO02 represents the second accession of Mombodica charantia collected in the Jugu district. From this activity, we have collected 1,412 accessions in 8 agroecological zones of Benin. The species collected include Amaranthus cruentus, Corchorus olitorius, Cucurbita maxima, Abelmoschus esculentus, Abelmoschus manihot, Mombodica charantia, Solanum macrocarpon, Solanum aethiopicum, Abelmoschus C.A.O, Amaranthus spinosus, Solanum americanum, Solanum nigrum, Citrulus lanatus, Gynandropsis gynandra, Amaranthus viridis, Lagenaria siceraria, and Solanum scabrum. The figure 2 presents the number of accessions collected per species. The highest number of accessions was collected for Solanum macrocarpon, Corchorus olitorius, Amaranthus cruentus, and Abelmoschus esculentus. Those species were considered as high potential for food and nutrition and were classified as top high national priority. Solanum scabrum, Solanum nigrum, and Solanum americanum recorded the lowest number of accessions collected. All the species collected have been recorded by Dancy et al. 2008, except Abelmoschus C.A.O, Abelmoschus manihot, Amaranthus viridis, and Cucumis melo. The figure 3 presents the number of accessions collected per species per agroecological zone. The central cotton zone and the northern cotton zone, both in the northern part of the country, recorded the highest number of accessions, while the area of fisheries and the depression zone recorded the lowest number of accessions. Moreover, all the species we have targeted were collected in the central cotton zone. The figure 4 presents the diversity of stem in okra species. There was a diversity in the color of a stem, in the shape of a leaf, and in the fruit in okra species. We have also recorded diversity in the leaf and inflorescence in Amaranthus cruentus. Diversity was also seen in the type of fruit in Mombodica charantia, in the color of the fruit. There was also diversity in terms of seeds, for instance, in Lagenaria, siceraria, and cucurbits. Regarding the seed sources, the figure 10 presents the seed source per species. Around 76% of the respondents used the seeds from the previous harvest. Moreover, Amaranthus spinosus and Mombodica charantia were found more wide. No seed were recorded from ATDA, and there was low seed paid from market. So the seed system is still informal. Figure 11 presents the uses of the collected accessions per species. The local communities use the species for multiple purposes, with the most common use as food. Abelmoschus, esculentus, Amaranthus, cruentus, Corchorus, olitorius, and Solanum macrocarpon were mostly used for food and sales purposes. According to Achigandako et al., they were the most widely traded. Some sister species were not cooked the same way. For instance, Abelmoschus manihot was creamed by hand, while Abelmoschus esculentus was cut by nick. Some other species were used for their medicinal properties, including Cucurbita maxima, Gynoropsis gynandra, Lagenaria, siceraria, and mostly Momordica charantia, which leaves can cure measles and vomiting, according to Dancy et al. 2008. Lagenaria siceraria is used for rituals, for sales, and to make art. Achigandako et al. 2008 reported its use as kitchen utensils and as musical instruments. Amaranthus spinosus and Amaranthus viridis were reported to be used as forage. In order to valorize these germplasm collected, some studies should be initialized, and these include studies to provide insights on the variability through morphological and molecular characterization of the accessions collected. Deeper ethnobotanical studies need to be done to understand the drivers of management of those traditional African vegetables across different socio-linguistic groups. The domestication process and appropriate agronomic practices need to be studied for some non-cultivated traditional African vegetables. The nutritional and the nutraceutical properties of the collected species needed to be deeply investigated. Investigation on the existence of natural interspecific hybridization must also be done. In conclusion, 1,412 accessions were collected from agroecological zone of Benin. The germplasm was stored at the genebank of the GBIOS laboratory and will be sent to the World Vegetable Center. The collected accessions were used for multiple purposes by local communities. Pertinent action must be taken to ensure the conservation and the improvement of those collected tabs. I would like to thank all the respondents that have provided seeds during this germplasm collection. The World Vegetable Center and MOUIZZ for funding this activity, and the GBIOS for conducting the survey. I could not finish my talk without wishing you happy International Year of Fruit and Vegetable. Thanks for your attention.